This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hello, everyone. Welcome to another episode of the Anthrobiology Podcast. Joining me on the show today, I have Dr. John Lindo of Emory University. How are you?
Very well.
Good. I'm so glad to hear it. I am really excited to talk to you because we're going to be talking about genetics. I was just internally laughing because I say I'm excited to talk to everyone, and I think that's true, but I am actually very excited to talk to you because you're doing some really interesting stuff in the lab, and you're also using math, which longtime listeners know I love math, and I strongly encourage every undergraduate to take statistics classes ASAP. So I think we're going to have a good time talking today. You have a few different projects going on in your lab. Can you tell us a little bit about them?
Sure. So I think one of the primary ones, like you correctly stated, was that I'm trying to really understand the genetic diversity of all the various populations that existed in the Americas, both North and South America, before and after European contact. So I'm also, in doing so, one of the overarching projects is to try to deconstruct this idea that there is one singular race of Native Americans, both North and South, and actually start talking about what the population diversity looked like in all these different regions and all these different ecologies that existed in North and South America that humans successfully inhabited and adapted to. And that's what I'm interested in. And that's one of the things that I think has been lost since European contact, since European colonialism sort of wiped away a lot of these evolutionary histories and certainly devastated many populations, eradicating some to 100%. So that's what I'm trying to rebuild. And and I do want to say that with a grain of salt, that I'm not trying to rebuild evolutionary histories for communities. What I'm actually trying to do is for communities that no longer exist, that were completely wiped out. And also for those of us that are in the United States, like myself, who identify as Latinx, my parents never spoke about anything about any type of indigenous relationship or anything like that. And so that, to me, is lost after European contact. So for me, I have no idea what relationship I have to some lost civilization from the Americas. And that, to me, is what I strive for in terms of putting eventually all of my work together comprehensively, and maybe one day allowing people who are curious about that lost indigenous ancestry that they may share, that they've discovered on 23andMe, how that might relate to some ancient civilization that has been discovered from an archaeological perspective, but also has genetic data that they can actually somehow relate themselves to.
Yeah.
So that's like one of the big overarching projects.
That's good. That's a lot. That's a huge amount to wrap your arms around. I think just some context for listeners who may not be as familiar with the history, especially of Central and South America and the Caribbean. There's been a lot of mixture, I guess would be the best word for it, because there are these huge native populations, right? And then you have tons of Europeans, especially Spaniards and the Portuguese showing up. But some other people too, like some English, some French, especially in the Caribbean. But then you also have effects of the African slave trade. And so you look at populations in Latin America, and it's really wild. It's just a really wild mix of people. In some ways, the indigenous populations in South America, especially in Central America, survived better than they did in North America.
Mm-hmm.
And you have elements of those cultures still existing as well. But like it's so wild. And it's so interesting that you're talking about like your parents didn't ever tell you really that much about indigenous cultures. My parents my dad's from Spain, but my mom's from Venezuela. And like my understanding of the indigenous culture in Venezuela is like they were very proud but warlike natives, and they were mostly wiped out because they were so warlike. And that's it. That's the extent that I know. And I never really thought about that till just now.
Yeah, I mean, I think it's for my parents, it was more something like they didn't even want to acknowledge that they might have any type of indigenous ancestry because of all the biases and bigotry that came over with colonialism in terms of how you identify.
For sure.
at least for immigrants into the United States.
For
But
sure.
that's I mean, that's contextual. That's going to be different for everyone. But
Mm,
that was my experience.
yeah, I know. And I think the idea of like the way that my parents especially talk about race, they immigrated here in 84 is always really interesting to me because they're like, well, we never really saw race in South America. And I'm like, I think you did, just not in the way that we see it necessarily in North America. Like it was a different experience,
Mhm.
but it's still something that's very much in the culture in different ways. Like you go to
Right.
Peru and the way that they talk about the indigenous well, the way that they used to talk about, I don't know what it's like now, the indigenous populations there, like there was a while they didn't even include them in the population count, which is crazy. They're like, they just
Mhm.
don't count.
Yeah, that's weird.
It's wild, you know? So it's like, it's still, especially with the indigenous groups, something that's really important to think about in the modern population. But we are here to talk about ancient indigenous groups,
Mhm.
um, which is, this is a really interesting topic, right? Like you're trying to suss out, like, what was it like in kind of this pre Columbian landscape? How do you go about doing that?
I think, well, a good example is for a paper is about to come out with respect to the indigenous people that existed in Uruguay before colonialism. Now, Uruguay is a, a sort of an outlier because this is a place where, due to military campaigns by the Spanish, where they intentionally eradicated the indigenous people there. And they're still there because of gene flow and the contemporaneous, I mean, in the modern population that exists there today. But in essence, those communities were wiped out in the 1800s. So we collaborated with archaeologists from Uruguay who looked at sites before colonialism, and they gave us individuals that were unearthed at these sites, and we were able to get their genomes. And for the first time, we were going to publish genomes of what the genetic diversity of indigenous people before colonialism, before they were wiped out, looked like. And also to potentially give some idea as or some relationship to the movement that's currently happening in Uruguay to re-identify as indigenous for those individuals that have that ancestry, and to actually have some sort of link back to the past before colonialism completely wiped them out. So that's I think a pretty key example as to how we're doing that. But along the way, then we through population genetic analysis, we find things that shouldn't be too surprising. Like we found an ancestry that hasn't been detected in South America before, but that shouldn't be surprising because we don't have too much. First of all, we don't have too many genomes from South America, period, living or ancient. And these are people who were wiped out. So we shouldn't expect that we know all of the ancestries that existed or all of genetic diversity that existed in South America before contact. We're really just sort of just at the point of really starting to, at the peak of the mountain right now in terms of trying to decipher this and trying to understand what the prehistories look like from a genetic perspective. So I think that's a good example in terms of how to do it.
Yeah, I think there's two really interesting lines of thought there. The first, let's go with the kind of archaeological bioanth one, which is that you're using essentially ancient DNA practices. So I think with that come a lot of the challenges that you get with ancient DNA, right? One is it can be hard to
Mhm.
extract ancient DNA, right? Like there's all the issues that always come with that, like contamination and then also small sample sizes. So I don't actually know what your sample sizes are. The article hasn't been published yet. But like when you're dealing with something like that, how do you proceed?
Right. So yeah, like you said, the first problem is trying to actually get any archaeological sample from an ancient individual that has any viable DNA. And in this particular case, in this paper, we only have two
Mm,
individuals. But luckily, though, is they yielded genomes, right? And so we actually, just with a sample size of one, because the genome is so big and because you're getting so much information from it, that can actually yield a tremendous amount for in terms of demographic analyses. You only actually really need one really nice genome to make incredible analyses such as different changes in population size, how they relate to other ancient samples and living, as well as you could even tell ancient migrations from only one individual because there's so much information in the genome. So it's 3.2 billion base pairs times two because we have two copies of our genome. So it's just one is all you need. But it's hard to get that super quality genome. But in this case, we got two. from two individuals before European contact.
That's really interesting. Can you kind of expand on that? Because one of the things that we've had whenever we have anyone who does paleo stuff come on is they complain about sample size. That's
Mhm.
like the constant undercurrent of complaint with anyone who does paleo stuff. How can you use genomes, even if it comes from just one individual, to look at whole populations like
Well,
that?
because one way to do it is you use windows. So you only use these windows only cover a certain section of the genome, and you can actually use that as data point. And by the time you cover the entire genome, you have many thousands, if not millions, of data points that you can utilize to then contrast this with other ancient individuals, other ancient populations, and other modern populations. So in reality, from a sample size perspective, it's actually quite large. And that's now, I will say that that's limited in terms of just making demographic analyses, seeing these relationships, population movements, population size changes. But where you do actually need a sample size is for things like detecting natural selection, where you need to estimate the allele frequency of an entire population. There, obviously, you would need something like 10 individuals to attempt some sort of estimation, and you couldn't rely on one.
That makes a lot of sense. I think it's the difference between being able to look at a single person's genome and being able to infer, for example, migration and things like that, because you're not just looking at that genome, you also have genomes from
Mhm.
other people, right? So
Right.
you can do kind of compare contrast across lots of different groups, even if they're not closely related. Like if you have like little bits, like you can still take a look versus like trying to determine something on a more individual level. Um, that's really, really interesting. That's really exciting.
The other cool thing about just using one genome is because your genome isn't a reflection of your parents. It's a reflection of, because of recombination and the way our genomes recombine before being sent off to the progeny. We actually have information of all of our ancestors going back basically hundreds of thousands of years as our species emerged from the ground. So that's even cooler. That's why there's so much information within one genome, is because of that recombination.
That's really cool. It sounds like there is so much data essentially to sort through, even just with one genome. How do you go about doing
Well,
that?
the only way that's been possible is because of technological advancements with both sequencing since around 2010, or I guess 2005, where we've been able to basically in parallel experiments where we can, instead of being relegated to only sequencing like 750 base pairs, all of these reactions happen at once so that now you can do a gigantic genome in a matter of hours. But then you also need the computational aspect, and that's been growing with it. Our ability to do all of these intensive computational analyses on very large data sets is really also, again, just become available in this century. And of course, it's getting more and more powerful. And all these methods continually change and attempt to improve each other. And they're academic, so they're all free. So it's a great business to be in if you need to use these type of analyses.
Yeah, I thought about that a lot when I was taking my stats course in grad school, because I was using a copy of SPSS to do to do my math. But I was like, I was sitting there thinking about like how long this would have taken to do by hand, and I was like, this would have been impossible. Like even just like the very basic intro to stats stuff that I was doing, it wouldn't have been impossible, but it would have taken me
Right.
months instead of
Yeah.
minutes.
I mean, and then one of the first steps that you have to do when we get the sequencing back is we have to align it to a reference genome. And that would be impossible to do that. Well, not
Mhm.
impossible, but I couldn't imagine the amount of time it would take to do that by hand to attempt an alignment. So,
Right. And the amount of error that you could
oh yeah,
potentially introduce as
absolutely.
well.
Although there are errors in the algorithm as
Mm.
well, but I'm sure that it would outperform a human.
Probably. So anyway, listeners, this is my bi-monthly reminder to take math and, start taking R as soon as you can. Actually, R is not as scary as it seems. It can be scary,
And
but it
it's
doesn't
free.
have to be, and there's lots of people out there and it's free. It's free and you don't have to do things to get it. You can just go to the internet. I'll put up the resources on my website just in case. But it's essentially just like a very basic programming language. And if you understand the math, which can be the tricky part, it's really not that hard ish. It's really not that hard at a basic level. Once you get We're not going to get into the mysteries of R. I'm sorry, that's not what this podcast is about. But do math, you'll like it. Slash, it'll be easier to get a job. Okay, so one of the other things that you touched on was indigenous communities or people, modern people who potentially are part of these indigenous communities who don't realize it, kind of like being able to find some of this via your analysis. That's a really interesting idea to me, and I think that like that really goes beyond just the science. Like, how do you mediate that genetic expression versus the culture that you grew up in that you really weren't a part of? Like, I think that those people are going to struggle with the same questions that people who might be discovering indigenous ancestry via DNA analysis might. There wasn't really a question in there, but it's something that I'm thinking about now.
Yeah, well, I think that's a complex identity issue, right? And it's completely contextual and up to the individual as to where they want to follow that. I think I myself am very interested in that loss aspect of my genome that is indigenous, but that might not be for everyone. Like you said, I'm also, American. I also grew up here in the United States from parents from immigrants, but I don't really identify with South America. I identify with the United States. So I don't think those identity questions are very complex, and I think everyone probably needs to grapple with them in some way at some point in their life, or maybe throughout their life.
Yeah, I also think about it a lot. For me too, it's they call it third culture, right? Because it's like you've got some of this Latin American stuff around here, and then you've got a ton of American stuff because you grew up here, and then you're like kind of just like this product of both. Like the other day, my mom called me and she's like, "Oh, Gabby, did you remember not to eat meat on Friday?" And I was like, "Because it's Good Friday." And I was just like, "I vegetarian." She's like, "Oh, right."
The bases are covered.
But it's like, and it's funny because I don't like, neither of my parents are particularly Catholic anymore, but like they still do some of these like cultural things, and then like it still extends to you. I don't know. I do think about this a lot, which is an example of how looking at these paleo questions or these ancient or even just historical questions can relate to the modern world. One of the other things that you're doing that you mentioned a little bit earlier was looking at allele frequencies. And that is a really interesting question to me, which is adaptation. One of the things you're looking at is adaptations that indigenous groups may have had to their environment pre-European contact. What kind of adaptations are you looking for, or are you hypothesizing exist, and how would you go about sussing them out?
One of the things that we were interested in, or at least when I was doing my dissertation, I was interested in this, sort of the narrative, at least when I was growing up, that Native Americans were wiped out due to colonialism because of the pathogens that they brought with them. And that basically also along with that, that essentially Native Americans just didn't have the immune system to fight off pathogens or something like that. And that didn't make any sense to me. So what I really wanted to do is try to go back before European contact and see if I could detect adaptation to pathogens, because there must have been pathogens here in the Americas, of course, that humans needed to adapt to in order to successfully inhabit both continents, which they did. And pathogens are one of the strongest sources of positive and negative selection or natural selection, period, in terms of humans and any organism. So I figured that that's a good way to start, is to look for signals of selection with respect to the immune system. And for my dissertation, that's exactly what we found. We found, of course, that one of the strongest signals of selection in indigenous populations that we were collaborating with in British Columbia, it was towards the immune system, indicating that when their ancestors arrived to British Columbia, there must have been some sort of, or some multiple pathogens likely that they had to adapt to, and they did successfully, presumably, to actually be able to inhabit there. So that's one of the ways that we started in terms of understanding natural selection in indigenous groups.
Yeah, that's really interesting. I think one of the things that people talk about a lot with, I don't know why, it's like in every intro to anthropology textbook, is the sorry, my brain just forgot the word for adaptation, is the adaptation
Mhm.
to altitude. Um, I think that that's one of the things that people talk about the most. Have you done any research on that, or are you hoping to?
Yeah. So for that one, where it's an ongoing project, but we did a project in the Andes in Peru around this area of Lake Titicaca. And there we were lucky enough to get really good ancient genomes before colonialism, enough to actually allow us to do a natural selection scan because we had enough to estimate allele frequencies. And the whole point of it was to utilize those genomes to try to understand the adaptation to high altitude. Because at Lake Titicaca, I forget how high it is, but it could be 3,200 meters, so very high, and certainly considered high altitude. But so the whole point of that was that perhaps these ancient genomes will reveal how they did it, right? Because unfortunately, the modern genomes that had been done previously in previous studies didn't show really strong signals that are associated with other areas of high altitude like Ethiopia and Tibet and Nepal. So I thought that maybe if I go before European colonialism, before the effects of the devastation and the population collapse, that I could find a stronger signal. So it turns out that I didn't. I did not find any of those hypoxia pathways or adaptation to low oxygen environments that you see in other areas of the world. Instead, the strongest signal of selection was to their culture. It was to a starch processing gene. And at first, that didn't make any sense to me. But then when I started thinking about it, it did make sense because the Andes is one of the great agricultural hubs of the world. And one of the things that they domesticated there were a variety of tubers, including the potato, as early as 6,500 years ago. So it makes sense that they also adapted to actually processing those starches in a more meaningful way for sustenance. And that's the signal that we picked up. So it was not an adaptation to high altitude, but an adaptation to culture.
That's super interesting. So, two things. One, you never know what you're going to find when you do research. And it's okay that you don't find like what your hypothesis is. Like, that's how you learn things. Like, that's so exciting. Um, two, the amount of potatoes in that region is insane. Like, you go and they're like, here is this potato. It's a crazy color and it doesn't really taste like potato. It tastes like some other thing. And you're like, incredible, delicious. Um, Third, I regularly camp up in the mountains here in California. And I will say that the only thing that really makes you feel better when you're feeling really gross is like sugar, it's carbs. So that actually makes sense to me. That's really interesting too, that you can see cultural adaptation in the genes. This is something that you worked on kind of tangentially. We were talking a little bit about before the show started, the gene culture coevolution framework, which is this idea that culture can have an effect on the genome, which it makes sense.
Yeah, it's a very strong source of selection, at least well in humans. Well, probably also in social animals as well. I'm sure that it exists there too. But yeah, it's a very strong source of selection.
For sure. I think that this is something that if people are interested in population genetics, it's worth reading up on. I can post the article where I learned about it that you were involved with as well. The other things that I saw in that article that were really interesting to me was I had no idea there's a few different projects that are super interesting, like the 1,000 Genomes Project and the Gene Tissue Expression Project. I had no idea that either of those existed, but they're basically these giant databases that collate genomes, and then also Gene Tissue Expression Project is it's exactly what it sounds like. It's the labels on the box. But I think that those are really, really interesting resources. And I think one of the things that I sometimes fall into a trap when I'm making this show is that the trap's the wrong word. There's a lot of really interesting people doing really interesting stuff out in the field. And I think that I maybe give the impression that the only way to do biological anthropology is by going out into the field and like sitting and watching a bunch of baboons for 8 hours, like for 40 days, or like working on a body farm or doing whatever. Like there's actually a lot of biological anthropology that happens in labs and on computers. And that's totally possible for you to do. Like you don't have to be someone who's like, I'm going to go and do a ton of field work and have to get a lot of vaccinations and maybe get parasites. Like you don't have to be that person.
No, I did try that out because I actually, when I went into my graduate school program, I actually wanted to be a primatologist. So I went into the field, and that was not for me at all. The whole thing where everything's trying to eat you or kill you was not appealing to me. But if you're into that, then the more power to you. I don't know how that's done. But yeah, so then I switched over to genetics shortly after that field experience.
I have to say, I think primatologists are really interesting people. They do really interesting research. I have never wanted to do it myself. I am extremely scared of other primates, including some human ones, but like all the non-human primates, I'm like, no, thank you.
Well, what about the little ones, like the tamarins? Those are adorable.
Huh. I guess they're probably okay, but again, they're in an environment where I'm good. I'm fine. I get upset when I see tarantulas when I go hiking here. I can't even imagine like the rainforest.
Mm-hmm. Yeah, yeah. Then that didn't gel with me.
Yeah, yeah, yeah. And I like the outdoors, just not that much outdoors. It turns out. Um, anyway, do you have any advice for people who are looking to get into the field? Into your field, not into the field as we discussed. Yeah.
Not a field.
Yeah, no, not a
My
field.
Oh, you mean on like on the graduate level? Like to go to like to start off and
Yeah, for sure. One of the things that we do talk about on the show is a lot of the times graduate school is not what people expected it to be. You know,
of course not.
yeah. So, and you know, like there are a lot of challenges, and there's like a lot of things that are changing in biological anthropology right now too. Like the field is changing, the kind of like the old guard is changing over. So I don't know, if you have any words of wisdom, feel free.
Well, I think to get into ancient DNA, it's a little difficult, mainly because there's only a handful of labs in the United States. But there's a lot of them in Europe, but that's a whole different game in terms of the way the master's and the PhD is structured. So I think it just in terms of any graduate school, the best thing to do is in your junior year, start to investigate what some labs that you might like and reach out to the PI and start gauging their interests. And also maybe they can give you some information about what their lab is like and things like that. But I think it's the first step is to reach out and start communicating with your potential PI.
Yeah.
To see if there's a good fit there.
Yeah.
But the other thing is I think before you go to any graduate program, well, first of all, it's hard to get in, right? But because there's usually like my program, we only have 2 spots every year for the biological side. So that sucks. It's just like we have to like a lot of excellent candidates don't make it through. So if you're lucky enough to get into a graduate program and you're lucky enough to have choices, then the next step is to contact the graduate students that are in the labs to talk about the culture and to talk about how the PI treats them. Because I think one of the bigger problems with being in a science lab is that you can be bullied and you can be treated like a brain on a stick and not a human. And that would be devastating. And unfortunately, I think that's more common than not. So I think that's a major issue. But that's probably like another podcast, right? To talk about being not treated like a human being in these science labs and things like that, either at the graduate or the postdoc level. So
Yeah. Yeah. That's definitely hard. And if you think that you can go to Europe and escape it, you can't. I ask you to refer either to my podcast with Dr. Tanya Smith or any of the podcasts that I did with graduate students who are all weirdly in Europe. I haven't had any American graduate students contact me to be on the show yet. But no, it's definitely a tough thing. I mean, even the rewinding to getting accepted, even that is really hard. The first time I applied to graduate school, I didn't get in anywhere. And I actually went and asked the head of a graduate program, Bernard Wood at GW. I was like, why didn't I get in? It's just very nice of him to let me into his office and explain to me why I didn't get in. And he looked at it and he's like, I honestly don't know. You're a great candidate. We just had someone who was slightly stronger. Like, you seem really nice though. which was a great response. And I did end up getting to do some work for him, and I got into graduate school the next year. But I mean, like, if you don't get in and this is what you really, really want to do, don't be discouraged. Like, there's always another application cycle, and there's ways that you can improve your resume before you apply again. But yeah, the bullying in academia it can be wild. And you're right. That's a totally another podcast. And I don't want to depress the youngins.
But if you do your research, you can avoid it, right? But presuming that you have choices, if you're lucky enough to have choices, then you can avoid it, because there are great PIs out there that encourage their students to be their best academic that they can be, or achieve whatever it is that they want.
Yeah.
So there are nurturing labs out there. You just have to find them.
Yeah, and I would encourage you, if you're really worried about it, to try and find a more established lab, because if like you end up in a lab where someone's brand new, there's just not that much information about them. Mm-hmm. Anyway, on that uplifting note, thank you for the advice. I always end the interview with the same 3 questions, which is I ask about books. So the first question is, if someone would like to know more about your area of research, what's a good place for them to start?
In terms of books?
In terms of books, articles, just any kind of resources.
Yeah, I would think for us, it would probably be like Nature review articles on ancient DNA. I think those are really comprehensive and well done, and speak to a broader audience. So I think like doing any of those Nature review articles is a good way to actually get a dive in pretty deeply, but in a comprehensible level, none of the jargon type of stuff.
Yeah, for sure. I think the other thing is, if you're interested in DNA, there is kind of a whole language that you have to learn before you get really deep into it. But you can get it by going into a high school or college textbook and just reading the sections on DNA. Like, it can be tough, but it doesn't have to be that hard. I'm reading a book right now, Human Diversity by Charles Murray, that does a really good job of laying some of that stuff out. And then the other thing that I really like, I just read a book. I want to say it's called Epigenetics. It's about epigenetics, which also does a really good job talking about epigenetics and gene expression, kind of like in a beginner way. I can't remember the name of the author right now, but that's also a potentially good place to start if you're just new to DNA. And I think epigenetics is going to be more and more important as time goes on. So,
absolutely.
Yeah. Sorry, that was your answer, and I was like, here's some more.
No, that's great.
Second question, what are you reading for work right now?
Yeah, I don't know. I'm always jumping from one journal article to the next. Right now, I think we're diving into some the genetics of skin pigmentation, because of one of my graduate students is working on an article, and we're actually examining the bias and potential white supremacy in some of these articles that only look at Eurocentric views about how skin pigmentation evolved. So that's sort of disturbing, but at the same time, it's being recognized, and we're trying to make an argument to incorporate ancient DNA to understand really how, actually look at evolution through time to see how skin pigmentation evolved in different geographies. So that's what the reading is in terms of what I'm doing now.
That's so interesting. And I love that you're tying in your area of research with also changing the field in an important way. That's really exciting. Also, skin pigmentation is I think it's something that's really interesting because it's something that's so visible to so many people. Mm-hmm. When I was teaching, that was always one of the questions that would come up, is like, why are people different colors? Like, why? How? I don't understand.
Yeah.
So I think it's such a good accessible thing for the general public too, which is exciting. Third question. What are you consuming for fun? I used to ask reading, but someone was like, I can't read for fun anymore. It's killing me. So what are you consuming for fun?
I actually agree with that. Finishing my 5th year as a professor, and I have not read anything for fun. It sort of kills that. I feel like to do something fun, I need to allow my brain to decompress and do something, quite frankly, mindless, which I enjoy. So what do I do for fun? Was that the question?
Yeah. I mean, what are you consuming? A lot of people go with like a TV show or a podcast. Some people still manage to read for fun, but not all of them. Someone said hiking the other day, which was really nice.
I don't know. I guess yeah, I do. I do like quality television, like series from HBO and things like that. Although I'll occasionally watch like the Netflix stuff, but like high quality, or what I consider high quality. Like I just
So
watched for
no
the third time,
Love is
like the Dark
Blind.
Crystal. Oh no, yeah, but I like sci-fi fantasy type of stuff. So I just keep watching Dark Crystal over and over. The series that they created, that Jim Henson's company created. I
Uh-huh.
sort of just think, well, blows me away how anyone can be that creative and have put that forward. So that type of stuff is how I decompress. If I'm lucky enough to have a series of it, it'll be something like that.
That's awesome. Just a note, not that I'm trying to pander, but like you are also very creative. Your research is incredibly creative. You're just creative differently than Jim Henson.
Well, if Jim Henson's company wants to hire me and to do something, I would leave academia.
You hear that? I don't think he's a listener, but you hear that, Jim Henson.
I don't think I have the skill sets for that.
It's actually really cool because I live pretty close to Pixar Studios.
Oh, cool.
Yeah, and I go climbing all the time, and I climb with this girl who does music at Pixar, and I'm like, what an incredible job. And then I tell her about my podcast, and she's like, how do you get to talk to such interesting people? So it goes both ways. Oh my gosh. But yeah, well, thank you so much for all of that. And I loved having you on. I really appreciate your time. You are welcome back whenever you'd like.
Yeah, thank you very much. This was very exciting. Thank you for having me on.
All right. Have a good day, everyone. Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show is produced by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.